Hello everyone and welcome back for more biotechnology. In this chapter we are going to start talking about some of the downstream processes that come after the transformation of bacteria or other cells with a gene of interest. Mainly our focus is going to be on identifying and purifying a protein of interest. So let's go ahead and start talking about this. So the first thing that we need to understand right off the bat is that even if you've identified a protein of interest, and even if your transformed cell is overexpressing this protein, meaning that it's making this protein in much higher quantities than most other proteins, there's still going to be other proteins in the cell. So a typical cell is going to produce thousands of different types of proteins other than just the one that you're interested in. So if you look at this SDS page gel here that's been stained with Kumasi, you can really appreciate our problem here. So if you look at the input lane, which is lane 2 here, lane 1 being the molecular weight markers, you can just appreciate how many different types of proteins there are. Some of them are high molecular weight and they run higher up in the gel. Some of them are low molecular weight and they run lower in the gel, and some of them are in between. But our conclusion here is that there are many different proteins that this cell makes. So if we just have a lysate from a cell, not only do we have our protein of interest, but we have a whole lot of other contaminating proteins. So oftentimes you will feel the need to purify your protein, meaning that you want to obtain a solution that contains as much of your protein of interest as possible and as little of those other proteins as you possibly can. But the conundrum we end up with is, how can we do that? How can we identify or even isolate a single type of protein when there are so many other different proteins present in the sample? So take GFP, for example. So at this point in the semester with our labs, we've already transformed bacteria with pGlow, and we have actually gotten cell lysates and run them out on an SDS page gel. And one of the exercises that we went through in lab is that we predicted, based on the fact that GFP has 238 amino acids, it should run at about 26 kilodaltons in an SDS page gel. But that does not necessarily mean that GFP is the only protein that will run at 26 kilodaltons. There is absolutely no rule that says that one protein can be the only thing that runs at that predicted migration pattern. So just because we get a band at 26 kilodaltons, that doesn't necessarily mean that it's GFP, and that doesn't even begin to help us come up with a strategy of how to get that GFP away from the other contaminating proteins. So you can hopefully appreciate the gravity of our problem here. But the good news is that it is absolutely possible to employ a number of different techniques and methods to purify and isolate our protein away from all those other proteins. And what we're going to end up doing is taking advantage of what we know about our protein of interest in terms of its physical properties, such as its size, how many amino acids it contains, its electrical properties, what's its isoelectric point, how hydrophilic or hydrophobic it is, what sorts of molecules does it like to interact with. So the more we know about our protein of interest, the easier a time we're going to have purifying it. So just to remind you of some things that we talked about in Chapter 8, we talked about the use of bioreactors like the one that you're seeing here, which are used to grow up very large-scale cultures of transformed cells for the purpose of making proteins. So we don't want to forget all of the work and labor that is involved in getting to this point. And then the other thing we do not want to forget is that if we are working with proteins, we want to work at cold temperatures. In this picture, you're looking at a refrigerated centrifuge, and during the purification process, oftentimes it will be necessary to centrifuge samples to separate soluble and insoluble fractions of cell debris away from each other. But refrigeration is necessary because you will recall from an earlier chapter that if there's anything we want to avoid when we're working with proteins, we want to avoid proteins becoming denatured, because at that point they are pretty much permanently damaged and then they are totally worthless to us. So the major thing that causes denaturation is excess heat. So working at cold temperatures helps us to avoid this. So very often when we're working with proteins, we try to devise our strategies and our methods such that we're always working at cold temperatures. Okay, so at this point we have our transformed cells. So let's go ahead and try to get our protein. Now we have two different ways of attacking this. The first is that we need to determine where is our protein in the cell. So most frequently when our transformed cells are making our protein of interest, that protein is still going to be inside the cell. But that's not always the case. Sometimes cells like to secrete proteins, meaning that the protein is made inside the cell and then it is exported out of the cell and placed in the extracellular fluid, or the fluid that surrounds the cell. So if this is the case, then lysing the cells is going to be totally unnecessary. So all we have to do is let our transformed cells make the protein, secrete it into the extracellular fluid in great quantities, and then at that point all we have to do is spin down the cells in a centrifuge and collect the supernatant, which is the extracellular fluid that contains our protein of interest. But like I said, much more often than not, our protein is still going to be somewhere inside the cell. So we're going to have to lyse the cell. We need to disrupt the cell membrane so that the protein inside the cell, along with everything else that we do not want, like DNA and membranes and things like that, everything else will leak out into the extracellular fluid, and then we can work from there. So cell lysis can be achieved in a number of different ways. One of the most common is by applying hypoosmotic stress. The idea here is that cells are incubated in a solution that will cause water to enter the cell by osmosis. The way this happens is that the lysis buffer that the cells are placed in has a higher water concentration than the fluid inside the cell, and this inequality in water concentration across the membrane causes water to want to move to correct that imbalance. So water enters the cell, and the cell starts to swell up until it ruptures due to the increased osmotic pressure. So that is certainly one way to go about it. Another common way that we've talked about is to use detergents, like sodium dodecyl sulfate is one that we've talked about before. So a detergent is going to poke holes in the cell membrane, and this is going to cause proteins to start leaking out. So sodium dodecyl sulfate is not typically used to lyse cells, but other detergents like Triton X100 or digitonin certainly are used. So the idea here is that you don't necessarily need to apply hypoosmotic stress. You can just include a detergent in your buffer, and that should disrupt the cell membrane to the point where the cell becomes lysed. Sonication is a physical or mechanical method that involves applying high-frequency sound waves to the cells, and this actually disrupts the cell membranes in much the same way that detergents do, except this is a physical method rather than a chemical one. And speaking of mechanical or physical methods, homogenization or French pressing is also a valid way to lyse cells. In this case, the cells are exposed to either high pressure or extremely high shearing forces, which will again destroy the cell membrane and cause the proteins inside the cell to come leaking out. So after you have your lysate, we run into the problem that we talked about before. Your lysate, the result of the homogenation or lysis process, is going to contain a whole bunch of different types of proteins, most of which you're not interested in. We want to get just our protein of interest. We don't really care about most of those other proteins. So one of the most common ways of purifying proteins is to use what is called chromatography. The picture that you're looking at here is a type of column chromatography, which we will talk about in the next section. So Typically what this is going to entail, and again in the next video we will talk about the principles and different methods of employing chromatography to purify proteins, but the general idea is that we take our crude protein sample, which again contains mostly undesirable proteins, and we pass it through a column that contains some type of binding resin. So you can see this binding resin here towards the bottom of the column. So this binding resin is going to interact with our protein of interest in such a way that most other proteins in the sample will not. So since our protein interacts with the resin and most other proteins do not, all those other proteins are going to pass through the column without interacting with the resin, while our protein is still comfortably bound to the resin. And then we can just use different methods to get our protein to fall off the column at a different point. Now, this is not necessarily a bulletproof way of doing this, so there will be some undesirable proteins that will co-purify with our protein of interest, but certainly this is a very, very, very good way at getting rid of most of those other undesirable proteins. Achieving 100% purity is probably not going to be possible, but we can certainly get a lot closer to 100% purity than we were before. And I mentioned this before, but there are different types of chromatography purification methods that we can employ, and each one is going to take advantage of a different physical property of our protein of interest, such as the molecular weight of the protein, its isoelectric point, its solubility in water, and etc., and etc. And we'll talk about all those different types of column chromatography in the next video. So the column that you saw on the previous slide is pretty simple. It's the most common and most simple type of column you can get, and it's relatively slow in terms of getting it to work, and that's because it relies on the force of gravity to draw the sample down through the column, so it's not working exactly very quickly. The column that you're looking at in this picture here uses a pressure pump to pull the sample down through the column. So this can be very useful because, again, biotechnology is a business and time is money. So you don't necessarily want to wait on gravity to get the job done when you can use a pneumatic pressure pump to pull the sample down and get things to happen much more quickly. Now, regardless of whether you're using a pump or not, something that is absolutely common to all types of chromatography is that you're going to be generating a lot of liquid waste. So in the bottom of this picture, you can actually see several different waste beakers. So you're going to be running a lot of buffer and a lot of sample through your column, so you're going to be generating a lot of liquid waste. So be prepared to collect a lot of that. So I mentioned this at the beginning of the video, but our focus in this chapter is what we call the downstream process, meaning everything that's happened once the cells have actually been successfully transformed, and we actually have established protocols for efficiently and effectively harvesting and purifying our protein. So we're really focusing on everything after the transformation process and after the protein has actually been made. So what our focus is we've already transformed the cells. The cells have made as much protein as we're going to want. Time to go get the protein. But the thing that we need to remember and what we need to take caution for is that we need to obtain protein products that are as pure and as concentrated as possible. So, like I said, it's easy to forget that biotechnology is a business, so we're not going to be doing good business if we are selling a protein product that is not very concentrated or not very pure. So we need to make sure that we're doing as good a job as possible during this downstream process. Alright, so that is going to do it for this video. As promised, our next video is going to start discussing principles of chromatography and how we can use chromatography to purify our protein of interest. I think you're really going to like that video. It's one of my absolute favorite things to talk about when we're talking about biotechnology. So I hope you'll join us next time, and I will see you later. Bye-bye.